Hello friends, welcome to the world of microbes, microbiology by Veerabhadraya Demaya. The world of microbes is very wonderful and it's very interesting. Comes under biochemistry. The human microbiome is the aggregate of all microbiota that reside on or within human tissues and biofluids along with the corresponding anatomical sites in which they reside. Let us have a brief look on anatomical sites of the human body. Microbiota, including the skin, mammary glands, placenta, seminal fluids, uterus, ovarian follicles, lung, saliva, oral mucosa, conjunctiva, biliary tract, gastrointestinal tract. Now, coming to the gut microbiome, which is having the inputs. It is having its own inputs depending upon the age, diet, demography, malnutrition, antibiotics, probiotics are all concerned with the gut microbiome. And the gut microbiome and the human genome is interrelated to each other. The interactions, genetic variations between gut microbiome and human genome. The human genome is related with the health and disease because of the microbes. So the microbes are harmful as well as they may not be harmful. Depends upon the nature of the microbes. This is all human microbiome. Let us pass on to the human microbiome. Types of human microbiota include bacteria, archaea, fungi, protists, and viruses. Though microanimals can also live on the human body, they are typically excluded from this definition. In the context of genomics, the term human microbiome is sometimes used to refer to the collective genomes of resident microorganisms. However, the term human metagenome has the same meaning. You please observe all these microbes which are living in our human body of different parts of the human body in one way or the another. Humans are colonized by many microorganisms. The traditional estimate is that average human body is inhabited by 10 times as many non-human cells as human cells. But more recent estimates have lowered that ratio to 3:1 or even to approximately the same number. Some microorganisms that colonize humans are commensal, that is, coexist without harming humans. Others have mutualistic relationship with their human hosts. See, these are the human hosts, microbes which are present in the human body in different parts of the human body. Human eats undercooked meat containing mature saccharocytes. These are the very important thing to know before going to cook the meat. You will get so many these things. So this is sporocyst, and which enters into the intestine, where many millions of microbes which are present in our body. Two types of bacteria, non-pathogenic and pathogenic. These are helpful or harmless. They have non-pathogenic are helpful or harmless. They have useful functions. That means they are in the decompose refuse, improve soil fertility, help metabolize food, protest against microorganisms, stimulate immune response. Saprophytes are monopathogenic bacteria that live in dead matter and do not produce diseases. Pathogenic, these are harmful disease producing when invading plant or animal tissue. Parasites requires living matter for their growth. Conversely, some pathogenic microorganisms can harm human hearts via metabolites. They produce like trimethylamine, which the human body converts to trimethylamine N nitro oxide via FMO3 mediated oxidation. Certain microorganisms perform tasks that are known to be useful to the human host, but the role of most of them is not well understood. Those that are expected to be present and that under normal circumstances do not cause diseases are sometimes deemed normal flora or normal microbiota. Human Microbiota Biome Project. The Human Microbiome Project took on the project of sequencing the genome of the human microbiota, focusing particularly on the microbiota that normally inhabit the skin, mouth, nose, digestive tract, and vagina. It reached a milestone in 2012 when it published its initial results. This is the emblem of the Human Microbiome Project, that is HMP. This is the DNA molecule, deoxyribonucleic molecule. This is a twisted ladder. We will discuss the biochemistry, my dear friends. Well, cellular living, which are parasites, protozoans, fungi, prokaryote, then virus and prion. These are the cellular living as well as the acellular non-living. These two comes under the acellular non-living, and these are the cellular living, that is parasites, protozoa, fungi, and prokaryotes. Well, introduction to human microbiome analysis. Human microbiome analysis is the study of microbial communities found in and on the human body. That means our human body consists of millions of microbes which are living in our human body in different parts of the human body. The goal of human microbiome studies is to understand the role of microbes' health and disease, traditionally studying samples from human skin, stool, or blood-related enzyme and labor intestine microbiology techniques of growing and isolating individual organisms, followed by phenotype or genotypic analysis. Microbial community profiling within a single sample was not possible with these methods. So, my dear friends, let us discuss about the important things. What do our microbes do? And there are 10 trillion human cells or 100 trillion microbes in the DNA molecule, as I discussed in the previous slide. You see here this microbiology, how stable the microbiome energies, that is microbiomes are stable, how long it will be stable. Around approximately every 6 months it may changes during the microbiomes. Now coming to the importance of chemical compounds which are present in our body. Reabsorbed bile salts are recycled by enterohepatic circulation. So these are higher science of microbiology, where the liver will have a function. The liver is bile salts, and cholesterol is there, and conjugation glycine, and this will, after the liver function, the gallbladder. This gallbladder is also function in the system, where the common bile duct will enter through this via the duodenum, and it will go to the small intestine, and it reaches the big intestine. During the process, 95% of the bile salts are reabsorbed by small intestine, and 5% of the bile salts are lost in feces. So these are the biochemical reactions which are taking place in our human body. So this is a important concept of biochemistry, the world of microbes, giving you the information from VBSN Academy. What is the microbiome, my dear friends? As I told you, it is started from this microbiome. This is from the stomach. There are 10 eggs, and it reaches so many microbes which are traveling along with the intestine. The Human Microbiome Project tells about all these informations. Gut mitochondria functions, influence of the immune maturation and homeostasis, host cell proliferation, vascularization, neurogenic signaling, pathogen burden, intestinal endocrine functions, bone density, energy, biogenesis. biosynthesis, vitamins, steroid hormones, and neurotransmitters, metabolism, branched chain and aromatic amino acids, dietary compounds, bile salts, drugs, and xenobiotics. Coming to the disease indications, neurologic, psychiatric, respiratory, cardiovascular, gastrointestinal, hepatic, autoimmune, metabolic, and oncologic. These are all the disease indications during the gut microbiota functions. Like, share, and subscribe, my dear friends. This is all about. I wanted to acknowledge this information taken from the internet and Google, then YouTube, Wikipedia. All these things are helpful to me for this given this information from VBSM Academy. I wanted to thank all the people of internet, Google, YouTube, Wikipedia, etc. So my dear friends, we will meet you in the next coming classes of biochemistry, the wonderful chemistry that is the world of microbes. Like, share, subscribe. Thank you.